Hello again. This is Dr. Dan Guerra from VerEvMed. Today I want to talk to you about pancreatic cancer. And to get into that discussion, we're going to also look at some of the specific aspects of metastasis. So let's go ahead and get started. So the title of this talk today is The Clinical Higher Ground in Pancreatic Ductal Adenocarcinoma, that we'll be calling PDAC, as the clinicians call it. Anyway, this image here I wanted to point out to you is on top of Pikes Peak, where I was just last month with my two sons. So you can actually drive up to the top of Pikes Peak and get the view that way, or you can take a railway system that goes all the way to the top, a cog railway system. And that's the route we chose. Anyways, great time. Recommend it to you if you ever get to Colorado Springs. So remember what VerEvMed is. We are scientists verifying published evidence in biochemical, physiological, pharmacological, and medical sciences. We are scientists. We are not physicians, and we're not pundits or lawyers. We are scientists. So let me get right into the grist of this talk. When we talk about cancer, we think about targets that can be drugged, for example, using pharmacological means. We also think about targets at the larger cellular level or even organ level when we think about surgery or radiation therapy. So we know that we're talking in pharmacology, then we're talking about targets for drugs. That's the realm of pharmacodynamics. So one of the druggable targets in many cancers are so-called receptor tyrosine kinases. These are basically enzymes which will add a phosphate to other proteins. So they're protein kinases. And the tyrosine means that's the place where the phosphate is added to, because tyrosine is an amino acid which has a free hydroxyl group which can be modified by a phosphate. So off and on phosphorylation of proteins regulates signal transduction pathways and even metabolic grids. So you can understand how that could be significant when we're talking about aberrations in those metabolic sequelae that could involve ultimately cell proliferation and cancer. So there are about 60 different isoforms of tyrosine kinases. They are all surface membrane bound, that is at the plasma lemma. They are almost always activated by combining with an extracellular ligand, and these can be of various chemical forms. And once that ligand binds, it induces an in situ, that means within the membrane, dimerization, or sometimes oligomerization, depending on the subtype or the subisoform. Mutations in the activation domain results in constitutive activity of these tyrosine kinases. And these are associated with several human diseases, not just cancers, but today we're talking about cancers. Also several inborn errors of metabolism, which, as you might guess, may have mutations in receptor tyrosine kinases. This is a huge field. So this paper that came out some 7 years ago from Cell, and there was the link there. This is open access. And I like this diagram because it basically shows you in very short order what could take me probably 10 minutes to say using words only. So you have various kinds of domains of proteins within plasma membrane. And those domains will be extracellular, they can be embedded in the membrane, or they can be intracellular, or that is usually cytoplasmic. So when we think about signal transduction, you can see how a receptor kinase is poised at a specific geographical location that is just unique and perfect for it to be able to transduce a signal from outside the cell to inside the cell. This is precisely what they do. They do it via, often as I said, binding to ligand and then dimerizing. Right? That means that you have two monomeric forms of the protein and they come together. Not necessarily coming together covalently, they just are reassociated in the membrane. So this gives you kind of an artist's depiction of that. These are various kinds of tyrosine kinases. There's TRK, KIT, there's the FGFR, there's the ERB. These are all actual big players in various forms of cancers. So you can see that here's nerve growth factor binding. Here are two subunits of this particular tyrosine kinase. You see how they're brought together to make this dimeric active form. Another motif can be in the stem cell factor binding to the KIT monomeric forms, generating this. These are all different subunits of the monomeric form. Here you have another one, FGFR. Again, this is pushing them together rather than keeping them associated in this sort of extended three-dimensional space. Here you have more proteins involved. So you've got the regular subunits, and then you've got these other subunits here. So this would be a heterotetramer of the receptor tyrosine kinase binding its ligand. So RTKs form dimers on ligand, which is the red shown here, binds extracellular domain. These are all outside the cell. The bound ligand stabilizes the separate monomeric receptor molecules. So it's that stabilization basically is an activation. And the activation energy that's transduced through the system. You also have dimerization, which activates another dimer, okay? So here you have insulin receptor, which is actually a tyrosine kinase. Here's KIT, and here's Ty2. Okay, so what you see, and these are disulfide bridges in between these two monomeric forms of these dimers. What you can do is induce via ligand binding, or sometimes just an alteration of disulfide bonding or phosphorylation on a residue between those two monomeric forms, and you make an active form, okay? And that active form is the receiver for a regular activator form of just a simple dimer. So it's dimer-dimer interaction. One dimer activates now the receiver, which has been modified here by phosphorylation, and now you have an active tyrosine kinase embedded again in the membrane. So you can see it's quite complicated. There are a lot of subunits here. There's a lot of molecular architecture that could be druggable when indeed you have a mutation in a tyrosine kinase. You can also see where various mutations, any one of which could cause a severe alteration of this orchestration of tyrosine kinase activation. Sorry for all the words here, but I want to get through this quickly today because spending more time than is necessary is usually not very efficient. So this is, again, this comes from a paper that's on open access that you can read. So we're going to talk about melanoma briefly here, okay, skin cancer. And melanoma is actually the third major cause of brain metastases. So the melanoma can metastasize to various locations in the body. And one of the places it metastasizes is the brain. So you end up getting brain cancer then. The other ones that metastasize quite well to the brain are, of course, lung and breast. Okay, so this is like what happens after you get the primary tumor. Then you get metastasis, and where that metastasizing can go is to other solid organs, but unfortunately also directly to the brain, which is usually quite lethal, quite quickly. So the management of melanoma brain metastases can be broadly divided. Okay, so I'm using this as an example. So how do you manage that? You manage the symptoms or you manage the etiology. Etiology means what caused it, okay? Or how the mechanism works to proliferate it. So the symptomology for brain cancer, what is used, what drugs are used are corticosteroids. Basically, they reduce inflammation and edema, so that decreases the amount of pressure in the brain, right? The swelling, that's what edema is. There are also anti-epileptics, which are used just to control seizure, because seizures are common in brain metastases. And there's also a pharmacotherapy directed just to maintain cognition. And these are, again, found in like neurochemical pathways. Now, if you want to attack etiology, it's a totally different type of drug and totally different type of mechanism. For one thing, there's surgery. Okay? If you are a candidate for surgery, sometimes they will do brain surgery and remove the metastasizing cell mass. There's also whole brain radiation therapy, WBRT, which is used. And then there's stereotactic radiosurgery, where there's a combination of surgery and radiation therapy. And increasingly, there's immunotherapy. And we talked about this in various other VerEvMed talks. These include activating T cells, which are otherwise quiescent because of suppression by foreign antigens produced by the cancer cells to suppress the specifically T cell response or immune response in general. There are also tyrosine kinase inhibitors. okay, and other kinds of protein kinase inhibitors, which are used even in brain metastasis. So there's all kinds of different armamentarium for this secondary result from a primary lesion, primary tumor. When we're talking immunotherapy, I've discussed this in various previous VerEvMed lectures. There's the CTL-associated antigen 4, and then there's a programmed death 1. When you antagonize these two receptor proteins, you can then activate otherwise suppressed T cells. This is acting on the T cell, not on the cancer cell. Okay, activate the T cell invades, it kills the cancer cell. It's basically what we're talking about. So for example, cytotoxic T cells or other kinds of T effector cells. All right, and those have been used for brain metastases. Now, here's a quote directly from the paper. 20% of patients with metastatic melanoma have brain metastases at diagnosis. So right at diagnosis, 1/5 of all people that come in that are recognized by their clinician, by the primary physician, primary care physician, to have melanoma, already 1/5 of them have brain metastases. You can see why this is really something you want to prevent entirely, or you want to get in there very early to diagnose this disease. So where does it occur? Where do metastases occur? Well, it depends on the cancer, but for this particular one, there are cerebral hemispheres involved. Probably 80% of all the brain metastases that come from melanoma are in the cerebral hemispheres. Okay? So it's a specific location. And that then will affect what kind of drugs you can use because of the pharmacodynamics, and also whether or not you can do surgery or stereotactic surgery or radiostereotactic surgery. All that is going to be related to the geography of the cancer. Okay. So what's the presentation? Again, just to get down to the clinical side a little bit, headache, which of course is common just about for anything, neurologic deficits, which again can be most monitored even by a primary physician, but certainly if you go to a neurologist, via an office visit. You also see deficits in neurological, in other words, just normal motor functions. And then there's cognitive impairment, of course, which is globally found in late-stage brain cancer. And then there are seizures, which also are very common. So if you get that cluster, right, that bad player cluster of headache, neurological deficits, cognitive impairment, and seizures, then they're going to be looking to see whether or not you have metastases from your primary cancer in the brain. So diagnosed melanoma brain metastases, really, really a tragic value here. The median overall survival is 6 months. So it's a pretty rapid decline and then death. 50% patients with advanced melanoma. Okay, if you're asking now what could be targetable here, we talked about some things. We're talking now about specifically a target in the kinase cascade pathway. About 50% of those who have melanoma that also get metastases in the brain harbor a BRAF mutation. And we're going to get back to talk about this protein. It's a BRAF protein. And it basically is a kinase that's downstream or distal to the receptor tyrosine kinase signal transduction cascade system, which can go awry in cancer. So here's here's BRAF, which is a proto-oncogene, Basically, it's a serine-threonine kinase, so it's not a tyrosine kinase. So tyrosine kinase is, again, phosphorylate tyrosine residues. Serine-threonine kinases, you get the OH group, the hydroxyl group phosphorylated on either the amino acid serine or the amino acid threonine. Okay. So this system physiologically transmits from extracellular signaling to the nucleus. So it's in between. Okay, it's in between that initial tyrosine kinase that we were talking about at the beginning, and then whatever happens downstream, which can be inactivation transcription factors, usually with some kind of phosphorylation cascade, like a continuation of adding phosphate to proteins, ultimately signaling inside the cell to trigger some kind of phenomenon. It can alter gene expression. It can also alter things like protein synthesis or protein degradation, autophagy, those kinds of things I've talked about before. So KRAS, big player. And KRAS is going to be, again, upstream. So KRAS, again, is a member of this pathway and it's associated with pancreatic cancer. So we're going to visit BRAF and KRAS together here pretty soon. These are two players. These are kinases that worked in tandem. And when one or the other of them is mutated, you can develop various forms of cancer. In this instance, now we're switching from melanoma, and we're going to start talking mostly about pancreatic cancer. Okay. I want you to get the idea, though, that the early etiology of the disease can often be a mutation in a gene that's common in various cancers. Okay. So what does KRAS do? It regulates proliferation, differentiation, migration, and apoptosis. And because it does, so does BRAF, because BRAF is downstream from KRAS. Okay. These proteins normally are essential for normal growth, particularly in utero development, okay, during gestation in humans and in mammals in general. Now, downstream BRAF, so BRAF is downstream from KRAS, is potentially oncogenic, and indeed it's druggable and targetable because it becomes an oncogene. It becomes a gene when mutated, starts causing tumorigenesis, and therefore it's implicated in many cancers. Okay. So here's a slide I got from Trends in Endocrinology and Metabolism paper published about 9 years ago. Now, what I want you to get out of this, I know there's a lot of information, and I know some of you don't have the background in knowing what we're looking at, but this is a plasma membrane. Those two lines delimit a plasma membrane. That's the outer membrane of the cell. So we're in that area where these tyrosine kinases function, and that's what this guy is here. Now, notice in this slide, I didn't show you the other half of this slide from the paper, but in this slide, you've got all of these proteins here associated with this tyrosine kinase. You've got RAS proteins, which are basically GTP activating proteins. Okay, so GTP, GDP bind to them. When GTP is bound to RAS, it's activated. When GDP is bound, it's quiescent, it's non-active. Okay, so in order to turn that system on, the tyrosine kinase has to work through the GRUB2, the SOS protein. Ultimately, GDP is replaced out with GTP, and then that induces this cascade. Okay, so I'm not showing you that. I'm showing you when you get the mutation. What's the mutation here? A valine at residue 600 of BRAF to an E. Now, what's an E? That's glutamic acid. Okay, so that's quite a shift, right? This is valine is a hydrophobic amino acid, and glutamate, right, is a hydrophilic amino acid. It's a big change, right? So, but it's only one mutation. If you look actually the coding for valine versus glutamate, it can only be a transversion from a pyrimidine to a purine, one nucleotide that codes for, from the triplet that codes for that amino acid shift. So all you need, and if you get it in that 600th residue, right, the code up at the 600th residue at the DNA level, you get a valine to glutamate, you get this mutation. Now, what does that do? That one simple mutation, it causes this entire system to become autonomous. It no longer is turned on by the normal signal transduction cascade. You don't have any ligand binding to the tyrosine kinase. You don't have any association with these other proteins in the plasma membrane, and you don't have any of this swapping out of GDP to GTP and the KRAS or any RAS protein, and therefore this is uncoupled from it. You get active BRAF. BRAF phosphorylates the MEK, the ERK, and then what happens is these ERK proteins, these are also kinases, okay? They break away from this complex because they're phosphorylated, and what they do is they phosphorylate a protein that is in the nucleus called ELK. ELK then phosphorylated binds to this SRF, which is part of the transcription factor system, which induces gene transcription, and what you get because of this lack of control is you get cells that are now transformed. Transformed cells means that they start proliferating. That means that this is the beginning of the oncogenic event that can lead to the pancreatic cancer. Okay, so that's the whole event. All it took was one nucleotide transversion. That means pyrimidine to purine or purine to pyrimidine. That's what a transversion is. In one particular codon for one particular amino acid, which made a valine to glutamate, and all of this goes terribly wrong, and you start getting cell transformation, cell proliferation, cancer. So let's look at this carcinomic setting here, okay? So you have the RAS-RAF pathway. Normally, you have a growth factor. It binds to the receptor tyrosine kinase, right? It's activated now because it's bound to GTP, okay? It then activates BRAF phosphorylates MEK, ERK, and you get normal cell proliferation and survival, okay? This is what normally happens in a good physiological setting. Now, in the carcinoma setting, what happens is you get, again, autonomous or constitutive activation. It's independent of any of this extracellular activation, okay? And it doesn't respond, therefore, to any back signaling, any back channeling, which all these things respond to normally, right? You always have feedback inhibition. Without any of this turning it on, this is just on all the time. Again, there's your one mutation, that valence 600 to a glutamate, for example. You you get the phosphorylation cascades going. You're just constitutive now. You get this cell proliferation, and plus the cells survive. So what do? They become cancer. Now, if you want to attack, that is pharmacodynamically target, you want to target BRAF, this is where you're going to do it. So there's selective binding of oncogenic BRAF kinase by one particular drug that's out there, VerEvMed, and that blocks the constitutively active pathway. So that's one inhibitor, which is specific for this BRAF mutation, which shuts this down. Okay, so that's a BRAF inhibitor, and those are out there in the clinic. So again, I'm trying to emphasize a clinical discussion today. Finally, let's talk frankly about PDAC. That's the most common pancreatic cancer. I think we all have heard of the fact that pancreatic cancer is quite deadly, right? It's difficult to target in general, pancreas is, and because of where the pancreas is located in the body. Again, geography is the word I use. Okay, or anatomy is probably the more appropriate term. So it's difficult to target the pancreas, and for you to be able to image the pancreas, you have to rely on imaging for the therapy. Before you even start in on doing any kind of therapy, a lot of imaging has to be done of the pancreas. Now, the pancreas has two basic components of it, right? It has an exocrine and an endocrine component. Now, we've all probably have heard of the endocrine component, even if we're not aware of it, if we're not medically trained, or if we didn't take biochemistry, cell physiology, et cetera, at university, or we didn't go to university. Doesn't matter. Just remember that the pancreas has two major mechanisms or functions. The endocrine, which we're not going to worry about today, involves insulin secretion as well as glucagon secretion. Remember what insulin does. Insulin is the hormone that allows cells to take up glucose, right? So, when a person is diabetic, they have sometimes a corruption of either insulin synthesis or insulin sensitivity. So the cells are no longer sensitive to insulin, and because of this insensitivity, you get type 2 diabetes, right? So that's the endocrine component of the pancreas. But this cancer is much more common to affect the exocrine. Now, what does the exocrine do? The exocrine, what that means is that it secretes proteins, okay, into the blood. So does the endocrine, but these proteins don't act as hormones. These proteins are actually enzymes, and they are, in fact, the enzymes that help digest dietary food intake. Okay. So the two main types of proteins that are made in the pancreas that we think about digestive enzymes are proteases and lipases. Proteases break down proteins from the diet. This is now going to be starting in the small intestine. And then the lipases, of course, break down dietary lipid, start making those chylomicrons, the resynthesis of a chylomicron and sending it into the liver for ultimately utilizing that lipid that you got from the diet to the rest of the body. Right? Protein doesn't require chylomicrons because it's broken down into amino acids. So anyways, the exocrine drains into the pancreatic duct, and that duct merges with the bile duct, the major bile duct, and that directly enters the small intestine. So you can see this is fundamental digestive process in the body, right? So you can understand if you get a carcinoma, if you get some kind of tumor in this exocrine portion, you're going to block all of this major necessary biological activity for nutrition, for being able to assimilate nutrients. All right. So because you're blocking or you're altering delivery of proteolytic and lipolytic enzymes if you get a blockage. So pancreatic cancer basically blocks off that bile duct. And first thing you notice, one of the first symptoms, presentations a patient may have, is jaundice, right? So they get jaundiced, right? The skin looks a little bit yellow because they're not completely metabolizing all of the amino acids, and they're getting a lot of urea in the blood. They're getting a lot of urine retention. They're becoming jaundiced. Okay, so this all ultimately leads to what's going on with the kidney because of the lack of appropriate digestion that's occurring from the bile duct being blocked, from the pancreas secreting lipases and proteases. So if we go further and we look deeper into the pancreas, the kind of cells we're talking about are called acinar cells. And basically, these are the cells that are in the exocrine pancreas. Okay, so they're different than the other kind of epithelia you get in the endocrine portion of the pancreas. Okay. So the acinar cells are basically ductal. They form ducts, right? And they are epithelial. So the most common pancreatic cancer invades this region and therefore the superior mesenteric artery and veins, okay, that are leading to the small intestine. Pancreatic ductal adenocarcinoma then is what we're talking about, okay? It's a ductal adenocarcinoma, PDAC, and it is the most deadly and certainly the most common form of pancreatic cancer. So it generates some of the etiology of the disease. It generates a fibroinflammatory response, okay? And that's going to be adjacent to the malignancy. So you're going to have cancer cells, then you're going to have all this fibrous tissue, which is also inflamed, which is adjacent to it, okay? But the cancer cells themselves are not in part of the inflammation process. They're protected, okay? So that's also important. That's why they're not destroyed by the immune response. So the bulk of the connective tissues, indeed, in pancreatic cancer are not cancerous themselves. They're fibroinflammatory. So genetically normal. These are not mutated cells. So the molecular profiling, trying to find out what molecules you might be able to target in the cancer in the pancreas, in the exocrine pancreas, becomes opaque or occult. It's more difficult to know what proteins are mutated in the cancer cells that could become druggable targets via the pharmacodynamic aspect of pharmacotherapy. So it ends up causing an immune suppression because you've got all this connective tissue, which is pro-inflammatory, but the cancer cells are not. So that it basically causes an immune suppression for the cancer cells at the actual carcinoma site, right, in situ. Therefore, there's a potential for immunotherapy, okay, because this is the situation. The way that pharmacologists look at this, the way the clinicians look at this, researchers look at this, is maybe we could do immunotherapy searching for whatever the actual antigens are in the acinar carcinomas. Okay, so those would be called antigens. And we're going to call them neoantigens because they are antigens made new dependent upon the proliferation, the mutation, right? So neo just means new, and the antigen just means something that will activate the immune response, right? Usually it's a protein. Sometimes it's a lipid. And it could be a carbohydrate associated with a protein, for example, self-surface glycoproteins. Turns out that many of the most potent antigens that we are talking about here in these cancers, and if you recall, we've talked about this just last time about hepatocellular carcinoma, very similar story there. Many of these neoantigens, the way that they work, if they do induce the immune response, is they have a particular molecular motif. And this molecular motif looks a lot like foreign invading microorganisms. Okay. Now, I used to teach molecular microbiology for many years, and what we call those proteins that microbes make that induce an immune response, we say they have, if they're pathogens, pathogen-associated molecular patterns. Okay. So that protein has a molecular pattern that is, there's a sequence of amino acids that in 3 dimensions forms a cluster, a structure, a globular structure of a protein that has a pattern, okay, has a motif, a 3-dimensional structure that is recognized by the immune response that triggers that immune response to kill those invading microbes. So those are called PAMPs, okay? Again, pathogen-associated molecular patterns. Now, interesting, right? So the antigens, the neoantigens that actually will trigger the immune response, and there are some that are made that do, but because of the suppression, they're not recognized because they're diluted out maybe, have very similar molecular patterns to things like invading viruses and invading bacteria that are pathogens. Okay. So that's a whole other story to consider in terms of evolution, including things like modern agricultural inventions some 10,000 years ago in humans, and then over time, the way our diet has changed, and also over time, the medicalization of the human race, removing parasites, preventing invading pathogens from occurring even before they do, like in immunizations. We are removing those molecular patterns, and in so doing, there's no longer a targeting of them. Cancer cells may be the source of this targeting if we can just amplify that signal, induce the immune response, and kill them in situ. Okay. So 2016 paper, this is basically one of the main papers I want to just, and again, I'm not showing you data today, I'm just trying to get down to the clinical aspects of it. This is a journal called Cancers. All right, so we know that pancreatic ductal adenocarcinoma is super deadly. About 7%, and this is the number worldwide, make it to 5 years. So upon diagnosis. So that's not a very good survival, right? So KRAS, that initial tyrosine kinase, right, the one that's linked to actually the tyrosine kinase that is bound to the ligand, all happening in the plasma membrane. Remember that slide. That's mutated in 95% of PDACs. And basically it's believed that that's what drives the disease, mutations in KRAS. So it'd be great if we could just target KRAS, right? However, potent and efficacious anti-RAS drugs in general are not well used yet because we haven't been able to isolate and determine what are good drugs. And so they're not in the clinic. Now, there are some drugs that are after KRAS, but again, they're in maybe phase 1, phase 2 trials, maybe very few of them in phase 3. So we're just now moving them through the pipeline. But unfortunately, having them have high efficacy, high potency, as well as high selectivity, combining those three attributes, right, making that three-dimensional corner in space seems to be somewhat difficult in terms of pharmaceutical sciences. So although drugs are being generated to knock out the KRAS, other alternatives are being considered. So those alternatives are what we call indirect strategies. And they include the RAS membrane association itself. So maybe if we can alter the RAS protein from even binding in the membrane, okay, maybe we could then cut that signal transduction cascade off, because remember that KRAS is mutated, it's generating this PDAC. Or maybe there are downstream effector molecule signaling. And we just saw that there are a lot of downstream signaling pathways, including BRAF. So besides the normal phosphorylation signal transduction cascade, which we looked at, okay, there are also other targets. And as a biochemist, I can tell you that those targets tend to be metabolic pathways, the kind of things you learn as an undergraduate or maybe as a graduate student in biochemistry, or if you haven't had that, you've probably heard of these things somewhere off in a distant canyon, right? So things like pathways such as glycolysis or glutaminolysis. Now, I've talked a lot about this in these VerEvMed lectures and in general in my 30 years as being a biochem professor. But glycolysis is basically the anaerobic breakdown of glucose to pyruvic acid. It happens in all cells. We've talked about it many times. Here it is as a potential target in this cancer. Okay, now we'll explain why in a minute. Glutaminolysis just means breaking down glutamine to glutamate and then using that carbon, right, to synthesize glucose. And that glucose then drives oncogenesis, right? So you need fuel to allow cells to proliferate, and so this becomes the fuel, this amino acid, okay? And that's common in many cancers, along with rampant anaerobic glycolysis. You also have other potential targets, mitochondrial ER-associated autophagies, again, something I've talked about at length in other lectures. I'm not going to talk about it further here because we're not going to bracket that in and look at it. And then, of course, the T-linked activating immunotherapy that I also talked about quite extensively in my last really long talk in hepatocellular carcinoma. Okay, so here's the link to be able to get this protein, to get this paper. Sorry. Okay, so what are we looking at here in terms of clinical approaches for PDAC? Well, surgery is probably the best thing, but when people are diagnosed, only about 20% of them or 50 of them are even eligible for it. Okay, so you may not be eligible for any kind of surgery for various reasons of complications that surgeons know. And so only 1/5 of the people that come in, they're diagnosed with PDAC, are going to be able to be available for surgery. And surgery just basically, it doesn't mean that you're going to stop the progression of the cancer completely, because the pancreas is a very regenerative organ, very highly biologically active. So surgery just gives you probably a longer time, not necessarily disease-free, but a longer extension of survival. Ultimately, radiation therapy, chemotherapy, as well as surgery are going to be used. And still, remember the 7% after 5 years, right? So that would be great if you can get surgery initially, and some people are candidates for that, and that is the first thing you're going to see if you're diagnosed with this or loved one is. But there's no cure from medical treatment available. But there are combined therapies. Okay, it's a very common motif in clinical oncology. So you hit them with something like a chemotherapeutic drug. So something like gemcitabine or something like cisplatin, the platinum-based chemotherapeutics. There's also combination chemo and radiation therapy. And those are largely for non-metastatic disease, because once it's metastatic, radiation is much more difficult to go after all those metastases, right? All those METs, they're called. So, but those are a couple of combinations that you get. Most people, of course, die eventually of metastatic complications from PDAC, even with prolonged medical treatment, surgery alone, or any of these combinations. Okay, so you have surgery, you have chemo, you have radiation therapy, and ultimately still 93, 95% of the people are dead within 5 years. And most of them are dead within 2 years. And if they have a brain metastasis, they're dead in 6 months. You get the point. All right, here's this, again, paper in Cancers, 2016, about a year and 4 or 5 months. So one of the targets is targeting, again, the whole T-cell, B-cell pathway. I'm not going to talk about that today. In this paper, they're calling them modulators of tumor microenvironment, looking at whether or not you can get immunoglobulin activity, macrophages, and maybe T-cells, okay? T-cells being, the effector T-cells being very potent when you can get them to work, to invade tumors. So that's one aspect. Here's the other possible targeting, looking at glycolysis, looking at energy production, right? Autophagy, right? Controlling autophagy, degrading or destroying some of those cellular components in the cell, or regulating that, continually kicking the cell back down to its basic components. Inhibitors of any of those metabolic targets, okay, do exist, okay? Then let's take a look at this. And I told you that if you can get KRAS never going to the membrane, there are inhibitors for membrane association. That's one druggable situation. You have direct inhibitors of the KRAS, preventing GTP from binding. And then you have inhibitors, again, here's the RAF pathway. I didn't even talk about the phosphatidylanosyl 1,3 kinase pathway. I did talk about P13K, AKT, mTOR in other talks. This is basically a protein synthetic pathway, okay? You want to knock that back, okay? You do not want that active in cancers. And here's the RAF pathway going to, again, all the way down to basically transcription factor induction or transcription of genes. All those genes being transcribed in the disease state that are going to cause cell proliferation are going to be bad players, because they're going to enhance the tumorigenesis. Okay. So there are lots of things to consider here, possible targets. See a lot of inhibitors of downstream signaling. So let's go back to look at BRAF. BRAF targeting is not as promising as one might guess, because you have subgenotypes in PDAC and in cancers in general. So there are 2 BRAF selective inhibitors that are out there, vemurafenib and dabrafenib, okay? Both of these inhibit, that's the IB part of the drug, okay? They inhibit BRAF, right? So remember that downstream from the KRAS. Cool. But guess what? Vemurafenib, which is very common in the clinic, is already approved for BRAF mutant melanoma. So if you get that mutation, for example, that valine to glutamic acid, V to E at 600, right? And this is in melanoma, okay? Big drug used in melanoma. And the response rate for this is about 50%. However, when a BRAF inhibitor is used when you have a background, okay, when you have a background subgenotype where you've got also a mutant RAS, right, which is further up that signal transduction cascade system, right? Paradoxically, what happens is you get an activation rather than inactivation of ERK, and then you get cell proliferation. So you go ahead, you add your vemurafenib, you're going after BRAF, you knock down BRAF. For some reason, the downstream kinase that BRAF is controlling, you knock out BRAF, but because you have a mutation in RAS, you're overriding, you're circling around, right? The BRAF targeting, the BRAF blockade, and you're overactivating the ERK pathway. You overactivate the ERK pathway, you even get more cell proliferation. Okay? These kind of paradoxical things happen all the time in clinical oncology. So what about the metabolic pathway? Okay, I can't not talk about this because I'm a biochemist, a metabolic biochemist. So this is really good stuff to understand. So PDAC cells show increased aerobic glycolysis and anaerobic glycolysis via the mutant KRAS. Now, how's that work? Well, the KRAS actually induces a signal transduction cascade that causes the expression of GLUT1, hexokinase, phosphofructokinase 1, and lactate dehydrogenase. Now, GLUT1 allows glucose to enter the cell, right? So that's fuel. Runs through glycolysis, right? Hexokinase phosphorylates the incoming glucose. Once you phosphorylate glucose to glucose 6-phosphate, it stays in the cell, gets metabolized. PFK is a rate-limiting step in glycolysis. PFK1 is, okay? And lactate dehydrogenase is down at the end of the pathway, okay? That's after pyruvic synthesis to make lactate. So any of those could be blockaded, right? Because all of those seem to be associated with this rampant cell proliferation. So there are non-selective genomic deletion of murine hexokinase isoform 2 target tumor cells. Okay, so if you get that deletion in that particular gene, you can slow down tumor growth. Okay. Also, if you use 3-bromopyruvate, that is a specific inhibitor of a hexokinase isoform 2. That does show efficacy in some animal cancer models. Okay? So again, this is subclinical or preclinical, looking at animal models, but it looks like they might be able to target some of these glycolytic systems, given this data. That's what we're looking at here. Interestingly, mutant KRAS also results in a drive towards non-oxidative arm of the pentose phosphate pathway. Now, you know why that is? Because if you're running glycolysis, you're making a lot of fructose 6-phosphate and a lot of glyceraldehyde 3-phosphate. Both of those two phosphorylating sugars can enter the second glucose utilization pathway endemic in all cells. And that's known as the oxidative pentose phosphate pathway, the OPP. Now, the OPP's main reason for existence, and again, this is being very teleological, it shares intermediates from glycolysis. So if you overload glycolysis by turning on all of these genes because of a mutant KRAS, you're going to flood glycolytic pathway. You're going to make excess fructose 6-phosphate, excess glyceraldehyde 3-phosphate. You can flood into the oxidative pentose phosphate shunt. That's the non-oxidative branch. Don't worry about what the oxidative branch is. We've talked about it in other lectures, and if you know what it is, and it's basically the production of NADPH and decarboxylation. There you go. Sorry. In any rate, non-oxidative branch just involves transaldolases and transketolases, alteration of the carbon number of different phosphorylated sugars. C4 sugars, C6 sugars, C7 sugars, and C3 sugars, respectively. Okay, so this is just altering the sugar repertoire so you can run those sugars back into glycolysis. Now, even though they're not directly associated with the oxidative branch, they are associated with making more C5. So you have C5 sugars like ribose 5-phosphate. Now, what is ribose 5-phosphate? Well, that is required for nucleic acid biosynthesis, right? So you know that you have nucleotide triphosphates, either deoxynucleotide triphosphates for DNA synthesis or just ribonucleotide triphosphates for RNA synthesis. So that means DNA replication, which is necessary for cell proliferation, and then gene expression, that is transcription, RNA synthesis. Both of those lead a lot of ribose 5-phosphate because these are ribonucleotides, right? That's the sugar that's in nucleotides. So that whole pathway then leads to more ribose 5-phosphate synthesis. So if you can blockade here, If you can blockade at that OPP pathway, or it's either called oxidative or just simply pentose phosphate pathway, doesn't matter. If you can blockade there, you can perhaps inhibit nucleotide biosynthesis. Therefore, you're going to deplete DNA replication, also gene expression. Okay, now what happens then? You're going to stop cell proliferation. At least you're going to tune it down. Amplitude signal modulation, tune it way down. You don't get a lot of nascent nucleic acid synthesis. You're not going to get cell proliferation, right? Because you're not going to get a new genome made every time you do a cell division. Yeah. So maybe that's important for those tumors that also have subgenotypes that are induced by DNA damage repair mutations. Remember the DNA damage repair systems I talked about at some length in 2 or 3 previous lectures, maybe 3 or 4 previous lectures? Okay. Well, the DNA damage repair mutations are another triggering phenomenon in carcinomas, in general, in any kind of oncogenic event, right? It's one of many. So if you've got a mutant in DNA repair, right, you're going to need to have a lot of nascent nucleotide synthesis because you're constantly repairing DNA. So if you already have a mutation in DNA repair and you don't provide adequate amounts of nucleotide so that you can repair, even though you have a deficiency in repair, the genome is still trying to repair itself. So less and less and less nucleotide to repair it, the cell will die. The cell will die, and it certainly won't divide because it can't make any more chromatin DNA, right? So you're completely out of the synthesis of those macromolecules, DNA, RNA, and so the cancer cell can't survive. Now that may be that could be useful, okay, knocking out the pentose phosphate pathway in conjunction with PARP inhibitors. Now, what PARP inhibitors are another new regimen of drugs that are targeting specific elements of the pathway associated with general oncogenesis. So what is PARP? Well, poly-ADP ribose. Now that particular nucleotide, adenosyl nucleotide sugar, notice ribose is in it, is necessary. You polymerize that structure is necessary for DNA repair. It actually links together during double-stranded or single-stranded breaks of DNA. You need that poly-ADP ribose as a substrate to link those two broken ends together while you're inserting the correct nucleotide, right, in base pair exchange or nucleotide excision repair, right, those two repair mechanisms. So PARP inhibitors are used. Maybe also we can get inhibitors of enzymes in the pentose phosphate pathway. You're going to decrease the amount of ribose 5-phosphate. You're going to make less ADP ribose. You're also going to be targeting an enzyme that polymerizes it to repair DNA. Wham, double negative for the cancer cell. It's going to die, right? Because it's not going to proliferate. All right. Here's the last paper we're going to talk about, brand new, out about a month. This is in clinical cancer research. All right. Now, this is a really interesting new approach. Okay, so we talked about metabolic grid, we talked about the KRAS, we talked about the BRAF, the ERK pathway. We were just talking there about combining them, maybe looking at nucleotide synthesis via inhibiting the pentose phosphate pathway. Maybe that linked to those mutations that also have as subgenotype the DNA repair mutations. Right? We just covered all of that quickly. Now let's talk about maybe a new approach. And we talked about the very beginning, so this is just looping back. So there are specific neoantigen subtypes that are made by cancer cells, particularly clinical researchers are looking at this in PDAC. Okay, remember I told you the main function of this talk is to get back to pancreatic cancer because it's so lethal and it's increasing in the population as one of the major cancers that causes death because we're getting better at curing or at least remitting breast cancer and lung cancer. Not that those aren't deadly, but we're getting better at remitting them, right? But we're no good at fixing pancreatic cancer, so it's increasing, right? All right. So, like the fifth element coming up and controlling the other four, that kind of thing. So, neoantigens, remember, are proteins that are made by the cancer cells. Okay, they're new, and they're antigens because usually the proteins are lipids, and they're new because the cancer cell is misfiring. It's making things it's not supposed to. It's making unique patterns. Remember what some of those patterns were? Correct. I hear you saying some of those patterns are pathogen-associated molecular patterns, the kind of things you get from viral and bacterial cellular associated material, right, such as proteins, glycoproteins, and even lipids. Right? So some neoantigens may be immunosuppressive, okay, outright, because even pathogens do this, and so do parasites. We talked about this in one of the previous talks as well. And those immunosuppressive neoantigens can mask others with a more potent antigen presenting, starting back at, let's say, the dendritic cell or the macrophage antigen presenting cell, presenting it via MHC complexes to the T cell. Okay, so the T cell becomes activated by a binary system. All right, so maybe these neoantigens that immunosuppress by blocking the more potent neoantigens that would otherwise turn on, say, cytotoxic T cell lineages or even any other T cell effector lineages. Okay. So perhaps if we can manipulate the T cell lineages with specific neoantigens, maybe as immunizations, right? We isolate what neoantigens are being made. We know which ones are suppressive. We try to block those, maybe with monoclonal antibody, and then we maybe then increase the population of the neoantigens, which activate via this T cell activation process via the antigen presenting cells. Those T cells, which will go out outright, invade that cancer mass, invade that tumor mass, and kill those tumor cells. So that's the idea. So that's what you want to do. You want to suppress the regulatory cells because regulatory cells themselves suppress the effector cells. So you want to maybe knock those down as a whole class of T cells. Tregs down, TFs up, TFs mobilized to the cancer site. Maybe that's a good inroad in getting a decrease in this morbidity and mostly mortality of PDAC. Okay, so neoantigens that activate T effector lineages appear to present as pathogen-associated molecular patterns. So again, those that seem to be potent, that are not the ones that are normally evading the immune and therefore suppressing the immune system, tend to be ones that have these molecular signatures or patterns that look a lot like bacteria and viruses, which are normally pathogens. Okay, again, this goes back to perhaps how mankind has shifted their diet, has shifted because of medicalization, what kinds of parasites and pathogens even get a foothold in the body, right? So the lack of those antigens are decreasing the T cell population, maybe making them anergenic, going into anergy, which means a sequestered senescent state. And maybe if we can amplify those neoantigens, because we already have a T cell population which is geared to recognize them because they have PAMPs, molecular patterns like pathogens. Maybe we can mobilize them, target that tumor, and kill it. That's the idea. And this comes out of this research group. I recommend you take a look at this paper. It's really good. In fact, I heard a recent podcast about this from a grand rounds. Anyway, manipulation of the immune response toward microbial pathogen mimicry, with my terms there, may be a new paradigm in clinical cancer research. Again, trying to look more like a microbial cell surface antigen, triggering that T cell response, getting that in situ right at the site of that tumor, particularly the PDAC, and knocking out that cancer cell mass, right? And evading or avoiding that whole immunosuppression that's caused by those fibrous inflammatory systems. All right, so hopefully that is quicker than I've normally given these talks, not as many slides, and I tried to be as efficient as possible. There's our email address, info@verevmed.com. You can also reach me at djgphd@gmail.com. Again, I'm Dr. Dan Guerra, and I'm co-founder and chief scientific officer of VerEvMed, and there's our website. And again, what we do with you as a client is we are scientists, okay? And we verify published evidence. So all the papers I talked about today are published. They're in that literature now. They've gone through the entire process of being published, right? We take a look at that evidence. We don't just say, well, here's evidence. We verify it, right? We carry out a hermeneutical analysis of that data. We try to see whether or not that fits into other research that's been done, right? And we try to see whether or not the data has been analyzed correctly, and maybe even the discussion geared in a way that looks most logical, right? Using categorical logic as our main thrust to study this body of literature. So we look at biochemistry, of course, physiology, we look at pharmacology, we look at microbiology, And we're putting all of those disciplines under the medical sciences, and we're trying to take a look at how the science then informs the medical science, and we're the scientist component of it. So give me an email, send me an IM, and I'd be glad to work with you. Anyways, have a good day, and we will see you soon.